Okay, so the mitochondria and the chloroplasts, these are going to be where cells get their energy from. And so the mitochondria is the site of cellular respiration, and the chloroplast is going to be where we do photosynthesis. So both of these are going to be enclosed by membranes, but they're not actually part of the endomembrane system. So instead, they're not enclosed by membranes, but what they do have, or they are enclosed by membranes in there, but they originated as a separate prokaryotic organism, is what is thought was their origin in there. So it's called the Endosymbiont Theory in there, that you had prokaryotes, and one of the prokaryotes engulfed another prokaryote that was able to do aerobic respiration. That one that was engulfed ended up staying in the other prokaryote and surviving and losing its ability to survive on its own. So that became the mitochondria. The reason they think that is the genetic information in the mitochondria in eukaryotic cells is the same as prokaryotic information in there with the genetic sequences, and there's a double membrane around it. With the chloroplasts, what is thought is they were a photosynthetic prokaryote that was engulfed by another prokaryote, ended up losing its ability to stay on its own, and ended up becoming the chloroplast in there. So that's all this picture here is showing you, is how these were engulfed to make things in there. So the peroxisomes, those are an oxidative organelle. Again, they're not part of the endomembrane system. They're enclosed by a membrane, but they are going to be used to oxidate things in there as a way for the cell to try and protect itself or break things down. So mitochondria are in nearly all eukaryotic cells. They have that double phospholipid bilayer around them. The inner membrane space is the region between the inner and outer membrane. And then the mitochondrial matrix is what's enclosed by the inner membrane in there. And that's going to have enzymes and DNA in there. It's highly folded in there. The more of those you have in a cell, the more it's able to do aerobic metabolism in there. So some of your muscle cells will actually have quite a few mitochondria in there to allow them to have better endurance and do more aerobic metabolism. So the chloroplasts in there are going to be a specialized member of the plastids in there. Some of the other plastids will store amyloplast, which is starch. Chloroplast is a pigment in there that's going to absorb UV light in there. So inside you have the thylakoids, which are interconnected membrane sacs. And they're going to form these stacks called granum. And then the fluid around that is the stroma. And then you'll also have DNA, ribosomes, and enzymes around the outside. So the peroxisomes are in here again, is one of the types. In there, they're going to be used in breakdown of fatty acids, detoxifying alcohol in the liver in there. And they produce hydrogen peroxide to do that. They're one of the tools to break down the garbage. We eat a lot of garbage, so we need a lot of tools. So the glyoxysomes, these are a specialized fat storing tissue in the plant seeds. So most seeds are going to be high in fat content. And that's because of the glyoxysomes. It stores the fat in there because when that seed starts to turn into a new little plant, it can convert that fat into sugars to feed that new little plant for a while. So it's just kind of, you know, a care package of food for that new little plant until it's able to start obtaining its own food. So your cytoskeleton, this is a fiber network in the cytoplasm. It helps to organize the structure of the cell. It provides mechanical support to make it a little bit stronger. It can interact with the motor proteins like your cilia and flagella. It provides pathways for things to move on. It helps to move the plasma so that you can do phagocytosis. And it also helps to regulate other biochemical activities that go on. What?
I'm just saying that's a very large flashcard. That's all.
Not necessarily. If it's not a flashcard you have to make. So your components of it, you've got microtubules, microfilaments, and intermediate filaments that are going to make up your cytoskeleton in there. The microtubule is the thickest, and it's made of the protein tubulin. It's going to help with the shape. It's going to help make the centrosome, which is going to be a region where microtubules grow out in animal cells. Within the centrosome, you have a centriole that is going to have 9 sets of triplet microtubules in a ring. And we know this is involved in animal cell replication, but you can have cells replicate without this because we can see it in plants or yeast cells. The centriole, we don't fully understand what it does yet, so that's why we kind of look at, okay, you don't need it in the plant cells, but the animal cells have this. So its role is not completely understood by a long shot. And this is just showing those 9 triplet rings, or 9 triplets in a ring. So your cilia and flagella, these are microtubules that are going to be extensions that project out from some of the cells. They help in propelling them. So flagella are going to have an undulating motion more like swimming. So they're going to move in the same direction as the axis. Everybody's probably seen on TV examples of sperm swimming. The cilia are going to work more like oars, and so you're going to have movement at a right angle in there. So with the flagella, tails in the back, it goes forward. With the cilia, they're going to be on the sides and move like oars to have things move forward in there. We have different types of cilia. The primary cilia are going to act like more of a signaling receiving antenna in there, so that things that go along in the outside of the cell's environment can trigger the cilia and pass the message on to the inside in there. We know that they're involved with brain function and embryological development in being able to see what's going on around the cell and determine how to develop from there. They have a couple different patterns in there. The 9+2 pattern means you've got 9 doublets and then 2 single microtubules in the center in there, and you're going to see that in nearly all eukaryotic flagella and motile cilia. The 9+0 pattern in there, you have the 9 doubles of microtubules, but you don't have the central pair, and you see that in the primary non-motile cilia in there. And then it gets anchored onto the cell with a basal body in there, so there's kind of a connection point anchor in there. Along the length of it, you've got these dyneins that are protruding motor proteins that are going to be responsible for the bending movement. So when ATP binds to those, they basically change shape. So they kind of move like this for changing shape. And that's what allows the sperm's tail to go like this and there. So this is showing it here, the cross-linking in there, and it bending. And this is showing it doing a wave-like motion. So in bacteria, it's a little bit different. Some of them will move in a corkscrew motion, but they use a different protein too. So the microfilaments are going to be smaller. These are solid rods made of the protein actin. It's a globular protein that you see in twisted chains. You'll have actin in your muscle fibers. It helps to support the cell's shape. It's going to help give it a consistent gel kind of consistency in there. It helps to bear tension forces so that the cell can stretch. It will be involved with the motility in there, so it allows amoeboid-like movements in there. So it can kind of scoot and wrap around and engulf things in there. It also is involved with cytoplasmic streaming, so it's a circular flow of the cytoplasm inside the cells. It basically keeps the cytoplasm moving inside the cell. And then your intermediate filaments are larger than the microfilaments. They're intermediate in size. They also help to bear tension forces. These will persist after the cell dies. They don't break down as easily. They help make up the nuclear lamina, lining the nuclear envelope to give it a little bit more strength in there. So the extracellular components, you have things that are outside the cell. You've got the cell wall in plants. It helps to protect the cell more, it maintains shape, and it's going to prevent excess water uptake. So for you as an animal, you can move around to control your water uptake a little bit. You can choose to drink. Even wild animals, they can choose to consume water, choose not to consume water to help regulate their water intake. As a plant, you're kind of at the mercy of what nature gives you. If it rains on you and there's lots of water around you, you need to be able to avoid taking up too much water or having your cells burst from taking up too much water. You also need to be better at preventing your loss of water, because if it doesn't rain for a long time, you can't walk somewhere to go get more water. So you'll also see cell walls in some of the prokaryotes in there, like the fungi and some of the protists too. So cell walls are thicker than the plasma membrane in there. You're going to have microfibrils made of cellulose that are embedded in the matrix. The matrix is basically polysaccharides and proteins. It will vary in content with the different organism. So primary cell wall will be what you'll find in a young plant, and it's a relatively thin, flexible wall in there. So if you look at new little plants when they're first coming up out of the ground, they're really flimsy. It's kind of the thing where if you water them, they fall over and fall back into the soil in there. And they just don't have very strong cell walls yet in there. It's going to have microtubules in the cortex that help to guide the cellulose synthase and orient the cellulose deposits so that it will affect the growth pattern in there. The middle lamella is the material between the primary cell walls of adjacent cells. So it's polysaccharide pectin in there that helps to glue the cells together. So it's a sticky material. So when you make jelly or jam, you use pectin that you can get from fruit in there. You can actually boil down some fruit matter and dry out the pectin from it to make your own pectin. It's not that expensive in the store to really justify the long process that it is to extract it. But pectin, when you add it to food, things like jelly, if you get jelly on your hands, it holds it together, but it also makes it really sticky in there. Yeah, we always used to joke because my mom's refrigerator would be so full of stuff, half of it rotten, that if you couldn't find a place to do it, you open the jar of jelly and spread some on the top of whatever container it is and stick it to the ceiling of one of the shelves to double the space. She doesn't really appreciate that joke about her refrigerator, but it's true. The secondary wall, this is going to be added between the plasma membrane and the primary cell wall in a mature cell, and it just helps to harden it more, make it stronger. So our cells need to be able to talk to the neighbors in there. So plant cells are going to have plasmodesmata that are perforated channels between the cell walls in there where they can connect to the neighboring cells. So this is showing some plasmodesmata in there. So on the outside of animal cells, you also have extracellular matrix in there. It's mostly glycoprotein. The most common one is collagen in there. You'll have some proteoglycans in there that are small core proteins with other carbohydrate chains that help to form a network that the collagen is embedded in. So in that picture you can see there's kind of the skinny little green fibers around it. And then you've got the purple that are the collagen fibers. They're a thinner fiber to help hold the collagen into place in there. And then the fibronectin is going to bind surface cell proteins called integrins in there. So you've got the fibronectins that are attaching here to the surface cell or the surface integrins. And the integrins are going to actually penetrate down into the membrane, into the cytoplasm in there. And it will attach to your cytoskeleton on the inside. So you've got this kind of web-like pattern on the outside that connects through these integrins to that web-like pattern on the inside in there. So that things that go on the outside, if it moves some of those feelers on the outside, it can actually transmit that message to the inside of the cell that way. So when we look at the types of intracellular junctions, the plasmodesmata are the ones that you're going to see in the plant cells in there. Tight junctions are one type you see in animal cells. That's what you're going to have in your skin in there to make it watertight. So if you spill water on your skin, it just sits on there. It also helps to keep you from evaporating as much. The desmosomes are going to attach muscle cells to each other in there. They're going to be made of intermediate filaments of keratin. And then gap junctions are going to be cytoplasmic channels that you're going to find in heart muscle. That's going to allow your heart muscle to have all of those cells contract together. So if your heart muscle cells didn't contract together and they were all just beating at their own rhythm, instead of the heart acting like a pump, it would just lay there and vibrate all over the place where everybody's doing what they want instead of working together. So these junctions in cells are really important. It's just easier to imagine what would happen in the heart if you didn't have that than other places. So this starts chapter 7 here. That's looking at the cell membrane in there. Cell membranes are going to have selective permeability. Some things can cross, some things cannot. And one of the reasons for that is it's amphibolic. It has hydrophobic and hydrophilic regions in there. Some parts like water, some don't in there. And you've got this phospholipid bilayer that helps to create this. So this is showing the phospholipid bilayer here. You've got your fatty acid tails in the middle, the phosphates on the edges. And this is just showing it enlarged in there. And our current model of cell membrane says it moves as a fluid mosaic in there. So the proteins can move around embedded within that layer. The easiest way I can explain it is if you think of that triangle when people play pool that they put all those balls in. If you leave out one of the balls, you can slide the other balls around and rearrange them in there. That's what your cell membrane is like. You can rearrange things in there. So that was not the original model. Back in 1915, they noticed that membranes from red blood cells had both lipids and proteins in them. In the 1960s, they came up with this sandwich theory in there, which they were like, okay, we know this phospholipid bilayer, and then they thought there was a protein layer like bread on the outside of that. And then in 1972, they proposed that instead of it being protein layers like two pieces of bread around a sandwich, that instead you had proteins that were actually embedded into the membrane in there. And that's the idea that is still accepted for it right now. So the membranes are fluid in that they can move around and change shape in there. Adjacent phospholipids can switch positions in there. They can switch positions 10 to the 7th times every second in there. So some of them are going to be slower. The bigger ones tend to be slower in there. Some are going to drift in a highly directed manner. Others are going to be attached to the cytoskeleton and not move around a whole lot. The temperature will affect them, so they remain fluid until the temperature drops and then they solidify in there. So if you get too cold, one of the things that happens is your membranes stop moving around and doing what they're supposed to. They're not as flexible in there. We have a wider temperature range that we stay liquid in there and are able to move around because of cholesterol in there. It acts as a temperature buffer. But that's part of the problem with getting frostbite in there, is that you've lost the fluidity and mobility of membranes in there, and the tissues start to die at that point. Once they're dead, they don't come back. You chop it off. Chop it off or it gets infected is pretty much what happens. So the membrane proteins and their functions, we have over 50 different kinds of proteins that have been identified so far in there in the plasma membrane of red blood cells alone. So to say we're going to know what all of the proteins are in there, at this point that becomes something where if somebody studies a special kind of protein on there, they become specialized in knowing a few of those. Nobody's going to expect you to go, these are all of the different proteins that are on the outside of the cell. We don't know. That list is so big that we likely will not ever know all of them. But some different types of them, the integral proteins are going to penetrate into the hydrophobic core of the lipid bilayer. where so if we have our lipid bilayer here, an integral protein is going to penetrate into it, where a transmembrane protein goes all the way through it. And then we have peripheral proteins that are not embedded, but they're actually attached to the outside in there as well. So the integrins are the integral proteins that are going to attach the fibers of the extracellular matrix. So these are the ones that are going to be attached to the extracellular matrix here. So things that the proteins on the plasma membrane do, they transport things. They'll be involved with enzymatic activities. They can conduct signals in there. Cell-to-cell recognition, so cells know who their neighbors are, helping to join cells together, and then attaching to the cytoskeleton and extracellular matrix. So in cell-to-cell recognition in here, you've got glycolipids on the outside that are short membrane carbohydrates that bond to the lipids, and glycoproteins, which is basically sugar and protein in there. And there's huge variations of these. There are a lot of markers for your cells. So you've got certain markers that are unique to you as an individual. You've got markers that are unique to you as a human that other species don't have. If you look at your immune system, that is how your immune system knows which cells are yours to leave alone. But it's also how some things will infect your cells in there. So if you get the flu in there, it's going to need a specific receptor on the outside, something like one of those proteins in there. And if it isn't there, it isn't able to get into your cell. So if you've been sick and you have pets and you're laying on the couch and you don't feel like getting up and going to the garbage, every time you blow your nose, you get the pile in there. And if your pets are anything like mine, they're like, oh, that's people stuff. It must be good. They go and eat it and things like that. But your dog doesn't get the flu. They don't have those receptors that you do in there for that. So that's why they can get away with it. There's actually certain receptors that are needed for the HIV virus, and there are a few people out there that are naturally immune to HIV because they don't have a receptor for it. However, we don't have a test to determine is that you, and you're far more likely to get hepatitis B if you decide I'm not going to get HIV, so I'm going to go hoe it up out there. You're probably going to get some other STD in the meantime. So. Yes. You don't have a receptor for it to get in and infect your cells. That's different in there. That's a different thing. But these people can't actually contract it because of that. But they're very few people. And it's still one of those things that just because you couldn't get HIV, do you really want to sleep with everything you come across? There's a whole list of other issues that goes with it beyond HIV for that behavior. So synthesis and sidedness of membranes. Membranes have a directional orientation. You can't just flip it around and go, well, the other side is just as good in there. They're going to be involved with synthesizing your endoplasmic reticulum, or they're synthesized in the endoplasmic reticulum and then get converted to these glycoproteins. So if you put the glycoprotein on the wrong side, it's not going to act the same. They get modified in the Golgi apparatus, which we know has a cis and transphase and in and an outside of it in there. So with our membrane structure that gives us the selective permeability in there, you've got your nonpolar hydrocarbons that are able to get through there pretty easily. Things like carbon dioxide and oxygen are hydrophobic, and they can cross easily because they're really small. So they can go in and out of the membrane anywhere. So if we make our membrane a little bit bigger here. So those can come and go through here in there. Some of your ions and polar molecules are going to be impeded a little bit more in there. So the things that are not able to get through, we have transport proteins that are going to be specifically permeable to specific substances in there. So some of your ions and polar molecules will have to go through a transport protein. So like saying, okay, you've got to take the bridge to get on and off the island. Some things, they can swim and go on and off the island wherever they want. Some are going to have to take the bridge in there. So the channel proteins are going to have a hydrophilic channel in there that certain ions will use in there. The aquaporins are ones that are specific to water going in and out. And then you've got carrier proteins in there that they're going to actually change the passenger as it goes through. So it comes in one way, and during the ride it gets changed and it exits another way. So you can think of it, you go on a bus ride, you meet some thugs, they beat you up, you leave with a broken arm. This picture just shows the different things that these proteins can do in there. Probably not the best place for the picture, I don't know why I put it there. So when things move across, passive transport allows substances to diffuse across the membrane and no energy is required. So when it says passive, that's telling you no energy is required. Diffusion, it just diffuses across anywhere, and then it's going to be controlled by the concentration gradient. So if you have really concentrated on one side of the membrane, less concentrated on the other, water wants to move to dilute it so that they are the same on both sides in there. So things will diffuse down its own concentration gradient. So here it's showing the molecules of dye and the water. Water is going to go this way, the molecules of dye go this way till you reach an equilibrium.
What does diffuse mean?
It means it just is able to float across and it doesn't need any energy. But it moves down its own concentration gradient. And this is showing with two different solutes in there that you can have them both going.
You said it floats across and it doesn't.
It doesn't need a transport protein in there. It would be passive transport if it needs that specific transporting protein. So osmosis will affect the water balance in here. This is showing how you have water selectively move across the membrane in here where you've got the higher concentration on one side and that the water is going to move over to this side here to try and dilute the sugar over here so that it's the same concentration here as it is here. In this case, those sugar molecules can't move across the membrane. So if the solid can't move across the membrane, the water has to move to do the job in there. So in cells without cell walls, they're going to be more susceptible to water changes in there. They can't tolerate excessive losses or gains in there. The cell wall is going to provide a little bit more protection in there. So tonicity is referred to as the ability of a solution to cause a cell to gain or lose water. Isotonic means there is equal on both sides. There's no movement of water. It's already equal. So hypertonic will lose water to its environment, which will shrivel up and die. Hypotonic, water is going to come in and the cell enters the cell faster than it leaves and it causes the cell to burst. So if you put water into a person's blood vessels in there where they've got red blood cells, there is more solids in the red blood cells than there is in the water around it. Water is going to rush into the red blood cells in there and cause them to burst because it's trying to dilute inside the red blood cell to be just like everything else. So that's why when you get an IV in there, it's in a saline base in there that's got some salts in there to help offset that. Otherwise, all the water would rush into your cells and you would die. So this is showing it in red blood cells in there. So if for some reason you're bleeding at home and you want to stick some of your cells into a cup of water, you can burst them if you really want to. With plants, they've got the cell wall in there, so what happens is this is in a normal state here. When it's plasmolysed, you can see that the cell membrane is starting to kind of pull away a little bit from the cell wall in there, so it starts to shrivel up. When you put it in the water in here, then it becomes turgid or filled up, and it's got the pressure against the side. So there's some plants that are a little bit more tolerant to you forgetting to water them. There's some daisies out there that you can actually get, and when you forget to water them, the flower kind of like flops over in there, and you can put water in there, and a couple hours later you go back and it's standing upright in there. And what you're seeing is the turgor in there, the cell's firmness, due to the inelastic walls exerting pressure back on it. So if you're pushed against the wall, you're going to be firm. If you're pulling away from the wall, it's going to make you flaccid in there. So facilitated diffusion or passive transport, this means it's going to be aided by proteins in there. This is when you have to cross at a specific location. Plain diffusion, anywhere in there. Facilitated diffusion or passive transport needs the protein in there. Still doesn't need any energy for it. So this is showing here that you can have the solid come in through the protein in there. This is showing it acting as a carrier protein where it comes in one way, exits another. So the gated channels in there, it's supposed to say gated, not graded. Here the channel proteins are ion channels that will open and close in response to a stimulus. So the most common example of these are your sodium channels in muscle. What's going to happen is it's going to take sodium in and push potassium out in there. And they're doing that all the time, and it pushes it out actually against a sodium-potassium gradient in there, so that when you allow it to open, it flows back in. You spend about a third of your resting energy just fueling sodium-potassium channels in your body every day. So we have glucose transporter proteins in there. They'll undergo a subtle change in shape to help translocate the solute binding site across the membrane, helping to move the glucose in. So active transport is going to use energy to move the solutes against their gradient. So you have to use a transport protein, but it's also going to have a little toll booth here on the side that it's going to have to fork over some energy for that to happen. So that sodium-potassium pump is one example of that. So here it's showing that inside your cell, you're trying to get the sodium out. So you move 3 sodiums to the outside. It has to bind an ATP to do that, your energy currency, and it puts them out on the other side. And then what it's going to do is it's going to take potassium from the outside and pump them onto the inside. So you have this one protein that moves one direction, the other in the opposite direction in there. It constantly does that. So when you do that, what you create is this voltage across the membrane in there, or the membrane potential. It's like an electrical voltage. You create a battery. It's essentially that you've created something like this across the membrane. So it creates an electrochemical gradient in there. Pump creates. So what will happen is once you have something comes up to open these channels, you can think of this being like teenage boys and girls. You've separated them. Boys are on that side of the hallway, girls are on this side of the hallway. They're at the doors clawing at them, trying to get to the other side because they want to be equal on both sides. What happens when you open one of the doors? Yes. They go for each other. They go off into corners. So that's what happens with the electrochemical gradient in there. So unlike with teenagers, though, in the cell, this is willing to do work to do that. If you can get work out of a teenager, good luck in there. So the electrogenic pump in animals is basically how you can harness that energy of allowing these to get to come together and use it to make other things in there. So the proton pump is the main electrogenic pump in there. In order for letting those come together, you're going to move hydrogens to the other side, and that's what allows you to make more ATP in there. So cotransport is an ATP-powered pump that's going to transport a specific solute, and it indirectly drives the active transport of other solutes. So here, while you're moving protons this way, it's indirectly allowing this one to pump sucrose this way. So cotransport just means you're having to have one thing go one way, another thing go the other way. Sometimes you're going to have things that need to go across the membrane that are bigger than just the molecules in there, that are a little bit larger. So when we want to do bulk transport of multiple stuff in there, that's when you're going to use endocytosis and exocytosis. So with exocytosis, if you're going to excrete some biological molecules in there, what you do is you basically push them up against the side of the membrane. It gets wrapped around the membrane and spit out in there. And that's what a lot of your secretory cells do. Yeah, sometimes it will just kind of make a membrane around it and it will leave in a vesicle. Other times it will just come through and the vesicle will burst and spit it out. So a lot of your secretory cells do this when you make insulin. A lot of your neurons do this when you make neurotransmitters. So if you've taken psychology, where you talk about how the neurons talk to each other and you have acetylcholine being released across, that's what it is doing there, is it's doing some exocytosis of the acetylcholine across the membrane. Endocytosis is like the opposite. It's when you take biological molecules or particles in and you form a new vesicle from the plasma membrane. So phagocytosis, phago means eating, cyto is cell. So this literally means cell eating. The cell is engulfing a particle. So it takes its pseudopodia and it wraps itself around it and engulfs the particle and takes it in. Once it's in, then it fuses with the vacuoles and the lysosomes break it down. If it's going to be droplets that it takes in, it's referred to as pinocytosis. A lot of times these are only distinguished a difference in a textbook, because when you're looking at cells underneath the microscope, especially a light microscope, you're looking at them in a liquid medium anyway. It all looks like a liquid. It's so small to us. But there is a distinction in there. So receptor-mediated endocytosis, this is a way of getting in bulk amounts of a substance even though they may not be concentrated in the extracellular area. So with this one, what you have are these receptor proteins in these coated pits in there, and it will form vesicles containing ligands that after the material is ingested, it gets released and the vesicle gets recycled in there. So by having the receptor in there, it's going to help to bring in more of it, and you can concentrate it and bring it in rather than having to just wait on the natural concentration to drive it. So we'll go ahead and take a couple minutes there, and then we might make it. The introduction to metabolism. So the introduction to metabolism. So the metabolism chapters are a couple ones that I'm going to recommend, even though you don't have the test on metabolism. You're going to want to look through those before we cover them in class, because metabolism is challenging. But the intro to metabolism is not too bad. It will be on this part, but there is chapter 9 and 10 are hard chapters. So I'm saying that even though your test is coming up on this, don't be in denial that those are coming. Read them and ease yourself into those. On 9 and 10. Yes. They'll be on the next test after this one. But a lot of times people don't look at the material when there's one test. They don't look at the upcoming stuff until they've got a test on it, and that's shooting yourself in the foot. So metabolism is how you transfer energy and matter, and it's subject to the laws of thermodynamics. Metabolism is basically just a chemical reaction that occurs in a living organism in there. It's just chemistry in something that's alive. And we organize things into metabolic pathways in there. These are reactions that are going to begin with a specific molecule. It will go through a series of steps and end up as something else at the end. And each step is going to be catalyzed by a specific enzyme along the way. And that's what we'll be doing in 9 and 10, is looking at these pathways. A catabolic pathway is one that breaks things down and releases energy. An anabolic pathway is going to consume energy to build things. So the way I remember this is I think cats are destructive. They break things down. Exactly. My sentiments exactly. So bioenergetics is the study of how energy flows through living organisms. So it's just the study of all of this stuff. So when we look at energy, energy is the capacity to cause change, and it can occur in different forms. Kinetic energy is the energy of motion. You can have heat or thermal energy, which is the energy associated of the movement of the atoms. Light is also a form of energy in there. Then we have potential energy, which is due to its location and structure. Chemical energy is the potential energy that's available for release in a chemical reaction. So in this picture, it's showing a diver being up high on a platform. He can release energy by going down. There's potential energy in his location there. When he's actually going down, he's got kinetic energy because he's in movement at that point. Now he's used up all his kinetic energy now that he hit the water. He's going to have to exert some kinetic energy to get back up onto the platform. So with chemical bonds in there, they're potential energy because you've got bonds of things being held together. When you break those bonds, the energy that was used to make them gets released in there with chemical energy. So the law of energy transfer, thermodynamics is just your study of energy transformation. And with systems, it's the matter under the study. Everything outside the system is its surroundings. So you can have an isolated system or an open system. An isolated system is unable to exchange matter or energy with the surroundings. It acts completely in isolation. Where an open system, you're going to have energy and matter that are going to interact with the surroundings around it. So do you think the human body is going to be an open or an isolated system? Open. You're going to interact with things around you. It's actually very hard to make an isolated system because there's air in rooms where we would study things. So it would have to be in a lab in most cases, a lot of times in a vacuum. It's just very difficult to isolate things from having potential interactions with even the air that's around them. So the first law of thermodynamics says that energy cannot be created nor destroyed, but it can be transferred and transformed. It's the law of conservation of energy in there. That basically it doesn't ever go away, it just changes what form it's in. And then the second law of thermodynamics states that during the transfer or transformation, some of it becomes unusable. So when you are transferring it, you didn't lose it, but some of it you can't use. And the way we usually have it end up unusable is as heat. And that heat gets released into the universe that makes it more disordered because you're putting more random energy out into the universe then at that point. So do those ideas make sense in there? First and second law of thermodynamics in there. So entropy is the measure of the disorder or randomness. It's the energy that gets transferred into the entropy of the universe. So how much energy gets lost to the universe into the random energy in there. Increased entropy is evident in physical disintegration of system organized structures in there. You can see that things will disintegrate over time in there. Spontaneous processes occur without energy input. So for something to be spontaneous, it occurs by itself. You don't have to put any energy into it. But for something to be spontaneous, it also has to increase the entropy of the universe. So it's releasing some energy, and you're always going to have some of it get released to the universe, which is increasing the entropy. So biological systems are going to increase the entropy of their surroundings in there. And one way that we can measure this is with the free energy. And it will be represented with the delta G on there. The delta G is because it's Gibbs free energy in there. And it's the portion of the energy that can perform work when the temperature and pressure are in a uniform system. So it's looking at how much can perform work versus how much can perform work and how much turns into entropy in there. So what delta G is actually derived from is the change in the system's enthalpy, the delta H, and the change in the system's entropy, the delta S, and the absolute temperature in kelvins in there. So this equation in here, this is just background knowledge for you of how they determine what delta G is. You can actually get tables in chemistry when you do this that tell you what the delta G is for all kinds of chemical reactions. So when you're trying to figure out, okay, is this going to be spontaneous or not, you just look on the table for what the delta G is in there. And you can figure out how much energy is released in a reaction in there. This is not something you typically calculate. It's more of the theory behind it in there. So there will be no math on this equation in here. So delta G must be negative for a process to be spontaneous in there. So delta G is your Gibbs free energy at the final state minus the initial state in there. So it has to be negative because you had to start with more energy at the initial state. And if you take away the bigger number in there, that's why you get a negative delta G on there. That part makes sense why it's got to be a negative delta G in there. So delta G is only negative when a process involves a loss of free energy. So when it moves to a lower delta G, it's a more stable state in there. So if you want to maintain a state of running around all crazy and everything, you have to have more energy to do that. As your energy dwindles down, what happens to you is you start to slow down and eventually sit down, maybe even lay down in there. So things just naturally progress to a state with a lower delta G in there. So this is showing here, he starts with a higher energy level. When he walks off the plank, he's going to naturally move to having a lower energy level. With diffusion, things start closer together. They naturally spread out into a more relaxed state in there. Same thing with the chemical reactions in there. So when we look at the free energy in metabolism, an exergonic reaction is one that's going to proceed with a net release of energy. It's going to release energy into the world, so it has a negative delta G. The magnitude of delta G represents how much the maximum amount of work that reaction can perform. There are things you can do to lower that. An endergonic reaction is going to be one that absorbs free energy from the surroundings. You have to put energy into it. And so it will not be a spontaneous reaction. It will have a positive delta G. So if you eat cookies and you're breaking down the glucose in your body, is that going to be exergonic or endergonic? Exergonic. It's going to release energy that you're going to have as calories, and if you don't burn them up, you'll turn it to fat. So is breaking down the glucose in those cookies going to have a negative delta G or a positive delta G? It's going to have a negative delta G because it's exergonic in there. Yep, that's when you're breaking down and getting the energy from the cookie. So with cellular respiration, this here, the C6H12O6 is glucose. You have glucose plus oxygen, gives you carbon dioxide and water in there. The negative delta G for this reaction is negative 686 kilocalories per mole in there, which is also, if you go with the other units, negative 2870 kilojoules per mole in there. So for each mole of glucose, 1 mole of glucose is 180 grams. That's broken down under standard conditions, which is 1 molarity, 25 degrees Celsius, pH 7. You have 686 kilocalories of available work that you can do from that. So if you need to do something that is going to require 1,200 kilocalories of energy, you would need to have 2 moles of glucose in there. The which one? Yes, it's similar to those that of how much energy it took, where you had the one for the kilocalories and stuff from last time. This is where this information comes from in here. This is, it's already calculated out for you in there. So of numbers that you would want to know in here, this one is one that you might see again in there. It's something you would likely see again in chemistry. It's not something you're going to see from me. Yeah, I mean, it's not something I'm going to test you on, but it's one of those things in chemistry you would see it again if you're working with delta G's in there, especially with biochemistry. So if you start out here in an exergonic reaction, you've got all the reactants. You're going to have some energy released and products. So you've got your products that you get at the end and the delta G. So these two together have to equal what this is in there. So some of it you got as products, some of it you got as delta G in there. It's like if you take in $20 and you ask for, can I get some coins and some change from it? What you get back may be in more pieces, but it's still $20 worth. So with an endergonic reaction, if you start out with these reactants and you want to end up with these products, this is how much work you're going to have to put into it in there. So it's kind of one of those things in the body when you look and people go, oh, well, you can eat this and you don't get any calories from it. For that to happen, you have to completely violate the laws of thermodynamics. There isn't anything that you eat in there that's food that has calories outside the body, but once you take it in, you don't. It's going to have calories in there. None of it, there's no food that you can eat that magically goes nowhere. Conservation of energy, it just doesn't disappear like that. So your metabolism is never at equilibrium because you're constantly having materials go into the cells and out of the cells. So if you're looking to get to this ultimate rest state, it never happens in cells. That's part of being alive, is that you're constantly doing metabolism in there. And usually you're going to have things get consumed that you metabolize in there, and you're going to take away one of the products in there, so you continually drive metabolism in one direction in there. So what's going to power the work is ATP. Chemical work is going to be something that pushes the endergonic reactions that are not spontaneous. You can have transport work that's going to pump something across a membrane against what it spontaneously moves as. Adenosine triphosphate. Mechanical work, that can be something like beating cilia, contracting skeletal muscle. And most of the time when you do work, you're going to use energy coupling in there. So you're going to use the energy derived from an exergonic process to fuel an endergonic process. You eat the cookies to get the energy to fuel you to walk across the room. And so they show it in a hydroelectric system here, that you're having the water go from the higher area to the lower area, and it spins this turbine and uses that energy to power the light bulb in there. So with these, if it's a closed system, once you get to equilibrium, it no longer does that. But if you have an open system where you continually take away this product in there, it just keeps running down there because you'll never fill this up. And a lot of times you break it down into multiple steps in here. And by breaking it down into multiple steps, you can direct the energy in different ways a lot easier. And the body is going to do that as well. When you break down glucose in your body, it's going to take multiple steps to do it, so you can take that energy and send it off to different places instead of getting it all in one big lump sum and saying, okay, here, now go split it up and fuel things. So the way we're going to transfer energy in the body and use as our currency is ATP, adenosine triphosphate. It's going to contain a ribose sugar, an adenine, and 3 phosphate groups on it. The bonds of the phosphate groups get broken by hydrolysis. And once the terminal phosphate or the third one's broken off, then it's ADP for diphosphate in there. So this is an example of your structure of ATP. You've got ribose, these phosphate groups, this adenine on there. So when you break off the phosphate in there, it's going to have a negative delta G of 7.3 kilocalories per mole under standard conditions. The thing is your cell is not standard conditions. In your cell is actually much more favorable than standard conditions. You'll get negative 13 kilocalories per mole. So under cellular conditions, ATP provides 78% more energy than it would under standard lab conditions. So ATP likes to work in our body in there. It's really happy to work in our body. So this is important because the energy that is released is somewhat greater than what most other molecules involved are. So you have to have that difference in energy in there to be able to drive reactions. So cells are going to harness the energy from ATP hydrolysis and use it to do work. So this is just showing where you have ATP with 3 phosphates. Hydrolysis, you put water into it, it's going to pull off one of the phosphates. Now you have ADP plus this energy. And this energy is what you use to do the work in there. And it's giving some examples here. For a transport protein, you're using the ATP to have the energy to transport this across a membrane. For moving this vesicle on this motor protein, it's giving the energy to scoot along the track in there. So in your cells, if you're using ATP to fuel everything, you're going to need to regenerate ATP. So ATP is needed to make everything move in your cells. You need it to make your muscles contract. Since you are using up this ATP, you have to get more, and the way we get more is through food. Food. That's what you eat food for. And your body likes to use carbohydrates as its preferred source of energy in there. You can get energy from other things, but it's not going to be as efficient at giving you the energy in there. So it takes ATP to make your brain run in there. If it's running low on ATP, it's not going to work all that well. So before you take a test, you really should have some food with some carbohydrate in it so that your brain can run better. So if you've been really hungry and tried to concentrate and make a decision in there, sometimes you're just like, oh, I just can't think right now. I'm hungry. And it's that your brain runs on glucose. Yeah, I mean, the carbo loading thing is kind of a... But that's, when you're out running, you're going to want to have mostly carbohydrate. that you replenish the system with. Yeah, it's the idea of loading. A lot of people look at it and go, oh, you eat the biggest pasta meal you can the night before. It's much more complicated than that if you want to do it right. It's to actually maximize the amount of glycogen stores in your muscle. You have to deplete them earlier in the week and go through this phase of being really uncomfortable and then replenish it in. But you don't want to put in so many calories that you've got it replenished and turning extra into fat stores. So it's kind of a delicate balance in there for it. And that balance just gets harder to do as you get older. In high school, you can get away with eating a lot of stuff because you have this metabolism that you don't realize how wonderful it is at the time until it's gone.
Well, in my sense, it kind of hurt me because I was always too small to play football, even though it's I was only a buck 80, so I could never gain weight no matter how much I did it. Now, years later.
You're like, where's that metabolism that I can eat whatever I want with again? You don't get a whole lot of carbohydrate from animal source, really. Yeah, there isn't a whole lot of carbohydrate in animal sources, so it only leaves plant sources in there. The smaller sugars you get it from it faster, but the faster you get it, the more the hormones spike up to take it in, and the faster you'll drop down in there. If it's the more complex carbohydrates that are more work to break down, you'll get more of a sustained release of it in there. But if you're hungry right now and you do that sustained release of it, you eat a whole bunch of it and you have no satisfaction because there's still the right now that wants it. So it's getting that balance in there. It's like when you say, oh, I'm really hungry and that candy bar looks really good, and you go eat a bowl of beans and you didn't put any cheese on it or anything, you just ate plain beans that are going to be really slow to break down. That candy bar is still going to look really good after eating that bowl of beans because you haven't gotten that sugar that your brain wants right now. So enzymes are going to help to speed up metabolic reactions. What they do is they lower the energy barrier. So a lot of reactions, you have a starting state that's a little bit higher, and at the end of the reaction, it's lower energy. But in the middle, you have to go over this hill. And that keeps these reactions from going on just uncontrolled all the time in there. If you have enzymes in there, you can control the rate of the reaction in there. So what it takes to get it over the hill is your activation energy. And then the transition state is going to be the summit of this graph in here, which is how much energy it takes to make them unstable so that the reaction can occur. What enzymes do is they act as a catalyst that helps to lower how much work it takes to get over this hill. So here, that was my little drawing of it that is jagged lines in there. This was one that I borrowed. You've got your reactants here, your products here, that there is a change in energy to get down there, but you have to get over this state here. And it's your energy of activation or your activation energy in there is what this represents. So some ways to get over that barrier in there if you heat up the reactions in there. So a lot of times when you put in heat into the system, you soften the bonds or you weaken the bonds in those chemicals that are held together in there, and it's easier to break them. So the activation energy lowers in there. pH can also do that in there, so if you change the pH of it, it may lower it. You can't do that with everything though, because you can denature proteins, and so you may say, well, okay, you know what, I want to speed up the reactions in my body, and if speeding me up to 99 degrees has me this much faster than 98 degrees, what if I speed me up to like 106? Well, at that point, you have started to damage your proteins and denature them. So there is a range in there where you can speed it up, but you can also just speed it up so much that you break it. So the enzymes don't actually change delta G, but what they do is they lower that activation energy barrier. So if you had to do this much work to get it started without an enzyme, with an enzyme, it only takes this much work. It makes it easier to get it started in there. So does that whole idea of activation energy make sense in there? It's kind of the hill you have to go over to get started. So when we look at enzymes, most of them are going to have the requirement of a substrate in there. And enzymes are going to be very specific about how they fit. You can't take an enzyme that's meant for one reaction and throw it in somewhere else and expect it to work. And they fit like a lock and key in there. So the substrate is the reactant the enzyme acts on. So here is your substrate. And it's got an active site in there, which is a restricted region of the enzyme that actually binds to the substrate. So a lot of times it's like this little pocket or groove. This goes in here and holds in this little space. And when it's together, we have the enzyme substrate complex in there. Most enzymes are going to have their name end in ASE. A lot of times if you have a sugar, its name ends in OSE. So if you're going to break down sucrose, the enzyme that does it is sucrase. Lactose requires lactase in there. So it's nice that the names kind of match up together in there. We can say they have an induced fit because once this is in there, it kind of snugs around it a little bit. So, you know, I can say, okay, yes, this tape fits under my arm, but I can induce the fit and hold it in there. And what it does is it brings things into position to enhance its ability to react. So what it may do is it may take two reactants, and when they come in, the enzyme brings them closer together so they can interact. So the substrate is held in the active site, and it will convert it into the product, and then the product departs the active site, and the enzyme is back there again. It doesn't get used up in there. So this can be very fast. A single enzyme can act on thousands of substrate molecules per second. Most are reversible, so the enzyme can catalyze the reaction forwards or backwards. It's just a lot of times you don't ever have a need to send the reaction backwards. So the way enzymes work, it can bring your reactants together in a proper orientation. So maybe they would normally come together like this, but the enzyme will bring them together in the reaction in the orientation they need to be to. It may help stretch them towards a transitional state. So it may go into that space and then it stretches it so it's weaker and able to break. It can provide a microenvironment that's more conducive to the reaction than the solution itself. So it may have a microenvironment in there where it changes the pH of it. It can direct participation of the active site in there where it may involve briefly bonding with the substrate and some of the R groups in there, and then it releases it again. So the rate at which your enzymes are going to function is going to be partially a function of the initial concentration of the substrate. So if you want to have the reaction start occurring faster, usually if you put in more of the substrate, it will start occurring faster. But it will have a maximum rate. Once you have filled up all of the enzymes, it can't go any faster. So it's kind of like if you're looking and saying, you know what, I want you to bring all these items from this table to the table over there. And you've got today to do it. If there's just one item on that table, you may look and go, I got all day, I'll do it right before I leave. However, if there is a whole big pile of things in there, you have to speed it up a little bit in there. However, you could make this pile mounds and mounds huge. There is going to be a maximum of how much you can do. You can only move so fast. And it's going to be the same thing with the enzymes. You can only expect them to do so much. They will reach a maximum rate. Once they're all full, it's full. You can't take any more. So enzyme activity a lot of times depends on temperature and pH because it will affect the three-dimensional structure of the chemical you're starting out with. pH is an easy way to change that, and temperature is an easy way to change that in there. So a lot of times enzymes are going to have an optimal temperature. So most human enzymes, their optimal temperature is at 37 degrees Celsius, your normal body temperature. Your optimal temperature for your thermophilic heat-tolerant bacteria is actually a little bit higher in there. So likewise, if you run a fever in your body, a lot of bacteria, they do have an optimal temperature that's right around yours in there. We know that you can be okay up to, right around in here. But things aren't going to happen as quickly for that bacteria if you do that. So with running a fever, what you do is you make a higher temperature that's going to be less efficient for them to do chemical reactions, but your immune cells actually function better at a slightly higher temperature. So what you're doing is turning down the rate at which they can multiply, turning up the rate at which you can fight them. And that's what your fever is doing. So with pH in there, there is an optimal pH for pepsin, the enzyme in your stomach. So if you take antacids, that enzyme is going to have a lot harder time getting to the optimal pH to digest your food in there, and a lot of times it won't get digested well. So cofactors are non-protein helpers for catalytic activities. A lot of cofactors are inorganic molecules such as metals and minerals in there. Your coenzymes are organic cofactors, and vitamins act as coenzymes in there to work with the enzymes. So where a person says, okay, I feel a lot better when I take B vitamins in there, I have a lot more energy. The B vitamins aren't actually giving you energy, but what they are is a cofactor or a coenzyme for the enzymes that you need to break down glucose in your body. So what they do is they let those normal reactions, they allow you to break down the sugar better, where if you're short on the vitamins, you may not have as many active enzymes, so the rate at which you can get the energy out of your food will slow down.
So what is your belief on multivitamins? So now in the media, they're saying that they have no use.
I think it depends on what a person's diet is. Because if you are getting all the vitamins you need to saturate all those enzymes, taking more isn't going to be useful. But there's a huge variation in what humans eat.
Because I know, like, for instance, with my wife, when she was pregnant, women have to take multivitamins.
They'll usually recommend it because people just don't necessarily eat a lot of foods in their natural condition, which is where you're going to get most of the vitamins in there. So your fresh fruits and vegetables, where most of those vitamins and minerals are going to come from. But when you think about it, there's a lot of people that haven't eaten an actual real piece of fruit or a real vegetable in years. That they only eat things that come out of packages. I mean, if you look at, I have friends that their husbands, it's like Hot Pockets when they're at home with their teenagers, Hot Pockets and frozen pizzas. There isn't a lot that is in its natural state in there. Yes, that's the reason that you see that with the vitamin D in there. So the things like vitamin D that we should be getting from sunlight, like today, yes, it's great that we have sunlight. Good luck going out there for long enough to expose some skin to actually have it do any good because it's so cold in there. So that's where a lot of those studies come from, is when they're taking people that are not getting what that optimal amount is. But we don't know what the optimal amount is for things. That's also the hard thing. And when you look and say, all right, this is how much you need to take today to compensate for what you didn't get in food, that's also really hard to know because we don't eat the same thing every day. You don't put out the same thing every day. So I think they're helpful for people that eat crummy. People who eat well, they're probably not making a huge difference for. And what you get from your food, we know when they're in their natural state, your body can use them a lot easier than when they're in a synthetic state. Unless your enzymes are short on it in there. So, I mean, sometimes there can be other reasons to do that, you know, like with B vitamins, they also cause you to put off a little bit of an odor in there that repels mosquitoes. You know, that can be a convenient reason to take them too. It makes it bright yellow in there. So sometimes the enzymes are going to have inhibitors. A competitive inhibitor is going to mimic the substrate, and it competes for the active site. So it's something else that gets in there and interferes with it. So this is another thing that could come into play. If you take something that's a competitive inhibitor for B vitamins into your body, maybe you're on a certain medication that will act like that, then you're not going to have energy because this is competing for where the B vitamins are in your enzymes. And with a competitive inhibitor, you have to try and overpower it more with more of the substrate in there. So it's kind of hard to offset that. Non-competitive inhibitors, these bind an enzyme in a site other than the active site, and then it alters the shape of the enzyme so that even if the substrate does bind it, it doesn't work. So a lot of toxins and poisons are actually irreversible enzyme inhibitors. Sarin nerve gas, it works by interfering with an enzyme. A lot of pesticides like DDT and parathion will do that. So when you look at, if you eat, I mean, we all have eaten, you know, on things that are not organic in there. If you go to a restaurant to eat, most likely they are buying cheapest produce, not organic produce in there. We're all taking in some of those things, and that's one of the theories in there that is, why are we seeing people be in worse health in there, is we're eating and taking in a lot of these things from our environment. Antibiotics may work that way as well. So there's other things that, even if you say, I don't eat junk food, you live in a world that's full of toxic things. Yeah, and you're also going to take in things from your environment without putting them in your mouth that can interfere with your enzymes.
I was in a doctor's office with a boy who was small, and he was talking like he was like, I don't know, like 2 years old. Oh, like, you're real smart. And they were like, he's almost 3, you know, like, oh, damn. And I was like, looking at him, and I was like, you didn't know that cows milk. They were like, the doctor's like, I'm not just thought we have to go to dairy farm and see how it grows. He was smaller, and I was like, because they're trying to lean, they tried to say that kids are trying to puberty earlier and all that kind of stuff due to what we eat that has a hormone.
Yeah.
This little boy's not getting milk or anything like that. He is super, super tiny. He's not big. I was like, that's weird.
There may be other issues in there, though, too.
No, his parents were tall. They were big people. I mean, he was just tiny. Maybe he was more tiny.
Yeah, you can't always just look at it and say with that, but a lot of what there are is a lot of things that will fit into an estrogen receptor in there. A lot of the antibiotics and hormones that you put in meat. If you give a chicken lots of estrogen, it gets fat. If you give a woman a lot of estrogen, she gets fat. If you give a man a lot of estrogen, he gets breasts. So, you know, we're seeing a lot more of those things happen because we put that in the meat. Yeah, you can kill a chicken and only keep it a third of the time that it takes to raise a normal chicken in there. So you make more money that way. But is it what's best for the person eating it? No. But it's what makes more money. But it is definitely with the kids. It's not unusual to see puberty happening at 8 years old. And it's just, especially for a girl, it's hard enough at 13 or 14 to start dealing with that estrogen and what it does to your brain. You take an 8-year-old's logic, and it's not good. So the allosteric regulation in there, This is where the protein functions at one site and it makes binding to it makes a regulatory molecule to a separate site in there. And you can use this to inhibit or stimulate things. So most enzymes are allosterically regulated in there. They'll have 2 or more subunits to it. We want to have these enzymes be regulated because they are very powerful in the body. If you have more regulatory steps, you have less chance of them accidentally getting turned on or off. So if you bind an activator to the regulatory site, it stabilizes the shape so that you can actually use the active site in there. So you can have fluctuations in the concentrations of the regulators in there that will influence the response of enzyme activity in there. And the substrate binding one active site may actually stimulate a catalysis of multiple subunits of enzymes in there. So they can act as kind of a cascade of events in there. You can have cooperativity in there where the substrate causing an induced fit in one subunit can trigger the same favorable shape in all other subunits of the enzymes. So it will allow you to amplify the response in there. In terms of identifying the allosteric regulators, it's really hard to identify these and isolate them to study them. We figure they're probably widespread in there. So the good thing about that for you as learning as a student is you get to learn about the class, not each specific enzyme. However, if you were to maybe go into like pharmacology or something like that, then they're going to study more enzymes more in depth because they're going to be looking to develop a chemical that can interfere with that. So that's when the need for the more detailed knowledge comes in. I think we'll go ahead and stop there today. That's actually quite a bit to absorb in there.